Hi, Will. How are you?
Good. Hello. Sorry for cutting it a little close.
Él empezó su carrera estudiando la interacción patógeno-microbio bajo la dirección del doctor Harcombe en la Universidad de La Trobe en Australia. Después empezó su doctorado en el laboratorio del doctor James Bull en la Universidad de Texas en Austin, donde empezó a estudiar un poco los fenómenos de ecología evolutiva. Y después combinó su experiencia de biología evolutiva con biología de sistemas en su postdoctorado en la Universidad de Harvard bajo la dirección del doctor Christopher Marx. Desde 2014, el doctor Harcombe es profesor asociado en la Universidad de Minnesota en el Colegio de Ciencias Biológicas, y del 2020 al presente ya es profesor asociado en la misma universidad. Las contribuciones del doctor Harcombe a la ciencia tienen que ver, son diferentes aspectos. Entre ellos está la predicción del impacto de antibióticos en las comunidades microbianas, el modelado del metabolismo a escala genómica, modelos de cocultivo y crossfeeding entre diferentes especies, y el impacto que tiene la estructura espacial en la dinámica de las comunidades microbianas. Y más recientemente también está estudiando la interacción de los bacteriófagos con las comunidades bacterianas. El día de hoy nos va a presentar su seminario titulado Bases metabólicas de la dinámica ecoevolutiva de sistemas microbianos. Will, thank you very much for accepting our invitation and to share with the students your knowledge and with us your research. Thank you for accepting and being here today.
Muchas gracias por invitarme. Lo siento que mi español no está suficiente bien a dar este seminario en español. pero yeah, es un gran placer hablar con ustedes. So I'll now switch to English, and I'm excited to talk with you all. I'm really disappointed not to be there, especially since we're in the midst of a snowstorm up here, and it sounds much nicer down there. But launching into things, my group is really excited about understanding microbial community dynamics. And so understanding the composition and the function of beautiful communities like the one I'm showing here, which is a community that was sampled from dental plaque, so from people's teeth. And you can see that there are a multitude of many species, that there's some really interesting spatial arrangement. And in addition to being both beautiful and fascinating, obviously microbial communities are really critical for everything from human health to global nutrient cycling. And there's also a lot of enthusiasm about trying to harness some of the power of microbial communities and diverse microbial assemblages for everything from crop production, so improving agriculture, to bioremediation, so cleaning up some of the pollutants in the environment. And so because of all of these important implications of microbial communities, we are really excited about trying to develop tools that will help us to understand the composition and function of communities, predict how those properties will change through time, and then ultimately also develop tools for managing the composition of these communities. So in our work, my lab is taking a bottom-up approach. And so ideally what we'd like to do is be able to start with a genome sequence and use that sequence to understand the physiology of individual microbes. So what they can eat, what interesting chemistry they can carry out, and how they might modify their environment. We then want to use this physiology of individual cells to understand something about how different cells or species may interact. And then ultimately, we'd like to scale up from these species interactions to understand emergent properties of the communities. We'd also like to understand how selection at each of these levels of biological organization feed back to change the genomes that encode life. And we tackle this integrative challenge through a combination of both experimental evolutionary ecology, using combinations of different modified strains in the lab, where we can track population dynamics and the mechanisms of interaction. And we pair that with a variety of different modeling approaches. One of those being a genome scale metabolic modeling that I'll touch on a little bit at the end of the talk today. So today I was hoping to share 3 brief stories about some of the ways that we're starting to think about how we can go from genome sequence to community properties. So I'll start with a short story about how metabolic interactions influence the effect of antibiotics. I'll touch on then how metabolic interactions influence the evolution of resistance, and finally touch on how metabolism can be used to predict community dynamics. So one of the questions that we were really interested in was whether crossfeeding might influence the effect of antibiotics. So crossfeeding is when one organism obtains metabolites from the secretion of another. So for example, this yellow bacteria may rely on amino acids or vitamins that are secreted by this blue bacteria. And we thought that these sorts of metabolic interactions might influence the effect of antibiotics, because you can imagine that if the blue bacteria is sensitive to the antibiotic, so it's inhibited by the antibiotic, while the yellow bacteria is resistant, that if these two bacteria are cross-feeding and reliant on each other, then you might get indirect inhibition of the yellow because it loses a partner upon which it depends. And if this sort of simple idea is true, that could be critical, because we are learning of more and more cross-feeding interactions between microbes in the human microbiome. So for example, a colleague of mine here at the University of Minnesota has shown that there is cross-feeding that occurs between anaerobes in the lungs of cystic fibrosis patients and a primary pathogen, Pseudomonas. Similarly, there have been well-documented diverse networks of microbes that cross-feed to degrade polysaccharides in our guts. So to test this idea of whether metabolic interactions are altering the impact of antibiotics, a really talented previous grad student of mine, Beth Adamovich, used an experimental system that we developed in the lab. So in this experimental system, we have a strain of E. coli, which is oxytrophic for the production of methionine. We have a Salmonella strain that secretes methionine, and we have a strain of bacteria called Methylobacterium extorquens that can secrete a nitrogen source. So we've engineered these three species to form an obligate mutualism when they're grown in lactose minimal media with methylamine as the nitrogen source. So the Methylobacterium provides nitrogen to the other two. The E. coli is the only one that can consume the primary carbon source, and therefore carbon byproducts are required by the other two from it. And then the Salmonella provides the amino acid, which is essential for the E. coli to grow. And one really nice thing about this cross-feeding system is that because we know what is being exchanged between each of these microbes, we can simply provide those metabolites in the media and then grow each of these strains independently under very similar physiological constraints. So they're consuming the same things. We're just changing whether they're getting those metabolites from the media or from the excretions of other bacteria. So Beth used this system and grew lawns of either monocultures or the tripartite co-culture on agar surfaces, and then put antibiotics on a filter disc in the middle of these lawns and measured the zone of inhibition. So very simply, we test the sensitivity of each of our monocultures against a gradient of antibiotics, and then also test the community. And as you can see, each of our three species had pretty different resistance profiles. So our E. coli and our Methylorubrum or Methylobacterium extorquens were pretty sensitive to the antibiotic and therefore inhibited at low concentrations of the antibiotic. But the Salmonella was highly resistant and grew right up to the antibiotic disc, which is here in the middle. And so the question then was, when we combine all three species, what pattern will we see? And it may be a little difficult to see on the screen, but in this case, all three species are inhibited at this low concentration of antibiotics. So even though the Salmonella is highly resistant, when it is reliant on these other species, it is inhibited at much lower concentrations of the antibiotic. And so we developed a very simple hypothesis based on this that we call the weakest link hypothesis, which is that when cells are engaged in obligate mutualism or obligate cross-feeding interactions, that the inhibition profile of all cells in that network will be set by the most sensitive cell. So the whole community will collapse down to the weakest link. We then were interested in testing whether this very simple idea, which we showed in a model system in the lab, had relevance in systems that are more important for human health. And so we collaborated with our colleague Ryan Hunter, who again has developed a really interesting cross-feeding system, or described a very interesting cross-feeding system between the pathogen Pseudomonas and anaerobes that exist in the lungs of cystic fibrosis patients. So these anaerobes degrade mucin, which is created by the human cells. And through degrading that mucin, the anaerobes release carbon sources, which the Pseudomonas can then consume. So we set up a very similar test. We first tested the inhibition profile of the Pseudomonas, and we found that even at our highest concentration of antibiotic, there was no significant reduction in the growth of the Pseudomonas. And this is consistent with what is often observed. One of the real challenges of this pathogen is that it is often really resistant to antibiotics and therefore can be difficult to treat. However, when we looked at the anaerobes, we saw a very different pattern, where now our lowest concentration of antibiotic dramatically reduced the abundance of the anaerobes. When we then tested a scenario, a cross-feeding scenario where the Pseudomonas was cross-feeding or was reliant on the anaerobes, we recapitulated our weakest link hypothesis, where now the Pseudomonas, the primary pathogen, is inhibited even at our lowest concentrations of antibiotics. Since this initial study, Dr. Hunter has gone on to test this idea with a whole array of frontline antibiotics and has demonstrated that across many different frontline treatments, that the response of the community to antibiotics is often very distinct from the response of individual strains. And one of the ideas that he's really putting forward based on this is that often in the clinic, clinicians will test the resistance profile of individual pathogens. But we often hear from clinicians that those tests are not very informative, that they are often not predictive of the effect of antibiotics in the patient themselves. And so he's suggesting that there's a need for developing better methods for testing antibiotic effects in a community context, and that may improve our ability to identify antibiotics, which will rescue some of our sickest patients. In addition to thinking about the level of inhibition, another really important thing for the response of bacteria to antibiotics is the death rate, or the number of persisters. So often what we see in the clinic is that we start with a population of bacteria, antibiotic is applied, there's then a reduction in the population size, but that there will be rare cells that are metabolically inactive that survive the antibiotic treatment. And then once the treatment stops, the pathogen population is able to rise once again to high densities. And so you can get these chronic waves of infections that continue to return. And so we were interested in whether cross-feeding might also impact not only the MIC, so the concentration at which microbes can grow, but also the rate at which they die. So to test this, another student in the lab, Sen Jiang, tested it with just our E. coli, so looked at the death rate of E. coli when grown in monoculture versus E. coli when it's grown in a cross-feeding co-culture with Salmonella. So to set up these experiments, the bacteria, E. coli or Salmonella, were each grown as monocultures and shaking flasks. And then either just the E. coli or a combination of the two were spread onto a filter membrane, or filtered through a filter membrane. This filter membrane was then put on a minimal media agar plate and allowed to equilibrate for 2 hours. The membrane was then moved to a second plate, which had 10 times the MIC concentration of ampicillin. And then we destructively sampled many different filters through time and measured the colony forming units so that we could measure the death rates. And one of the things we found is that we found nearly 100-fold more persisters when E. coli is cross-feeding. So the x-axis here is time in hours, the y-axis is fraction surviving. You can see that when we put the bacteria onto antibiotics, they rapidly start to die off. The blue line here is the E. coli in monoculture, and the green line is the E. coli in co-culture. And you can see that initially, E. coli has very similar death rates in both monoculture and co-culture. However, we see that then there's a stark change in the slope of this death curve, such that significantly more E. coli survive when they're in a co-culture treatment relative to a monoculture treatment. We were interested in whether this effect was driven purely by cross-feeding, or maybe just by the presence of Salmonella. Maybe Salmonella was releasing a chemical that altered the frequency of persisters in our E. coli population. However, we ruled this out by repeating this test, but in a liquid environment. So if we do exactly this test, but grow the bacteria in liquid and then treat them with antibiotics in liquid instead of on these filter membranes, we see that now this difference in survival disappears. So now E. coli has the same survival rate in both monoculture and co-culture. So it seems that this difference has something to do with spatial structure. And our intuition is that what is happening is that when cells are growing on a surface, that there is an added level of stochasticity in how close an E. coli lands to a salmonella. And that what that does is it increases the variance between cells. It increases the heterogeneity of cells, so that there's slight differences in the mean, but that most importantly, you see sort of a bigger spread in how rapidly cells are growing. This is ongoing work, and so we're currently struggling with some microscopy to definitively demonstrate this. But it seems to be the case that there is something about the variance that is driving this effect. And I'll highlight that this has some similarities to some of the beautiful work by Dr. Pena-Miller. However, in his work, the variance was driven by plasmids, whereas in ours, it seems to be driven by something about the interaction with other species. So in summary of this first part, I've shown that cross-feeding makes it easier to inhibit bacteria, that targeting cross-feeding is effective in health-relevant systems, and that cross-feeding makes it harder to eliminate bacteria. So moving on to a second story on whether metabolic interactions influence the evolution of resistance as opposed to just the ecological impact. So based on our initial results, we thought that cross-feeding might alter the rate of evolution. So we first set up a simple mathematical simulation in which we set up a series of habitats, and we simulated an increasing concentration of antibiotic across these habitats. We then inoculated bacteria and simulated the growth of, simulated growth within a single habitat. And each time that the bacteria divided, we sampled from a distribution of mutations such that some bacteria picked up resistance mutations. After a period of growth, we would then subsample from a given population into a fresh environment that had the same antibiotic concentration and a higher concentration of antibiotic. We eliminated any cells that did not have sufficient resistance to survive in that environment, and then repeated the growth process. And we did this in simulations with either just a single microbe or a co-culture, which followed the weakest link constraints, such that microbes were not able to grow in a patch if their partner was not also present in that patch. And what we found is that if we had just monocultures or 2 or 3 species cross-feeding co-cultures, in all cases, we saw that the minimum inhibitory concentration increased over time. However, you can see that the rate at which resistance increases is much faster if cells are growing as monocultures versus if they're growing with other species on which they rely. So we had this very simple model. We were then interested in experimentally testing it. So we went back to the lab and again used either monocultures of E. coli, monocultures of Salmonella, or a co-culture cross-feeding between these two. And we, as with the mathematical model, we set up a gradient of antibiotics, in this case along a 96-well plate. So we would inoculate either E. coli, Salmonella, or a co-culture along an entire well. They would initially be able to grow up to some concentration. And then we transfer these plates every 48 hours for 10 to 20 transfers. And our transfer regime mimicked that in our model, where after 24 hours, we sample cells from one well, put it into a new well with the same antibiotic concentration, as well as one antibiotic concentration higher. The numbers in these wells are meant to represent the concentration of the antibiotic. And we did this for gradients of both rifampicin and ampicillin. And what we found was that, consistent with our model, in co-culture, populations evolved resistance much more slowly than in monoculture. So again, what I'm showing you is time on the x-axis and a measure of the resistance of the, in this case, E. coli to rifampicin. And you can see that across many replicates, that there was significantly greater increase in the resistance profile of E. coli when it was growing alone versus when it was growing in co-culture. And this pattern held if we focused on the salmonella. So we compared the salmonella in monoculture to co-culture, and also replicated across drugs. So this seems to be a very strong and robust pattern, which is highly repeatable. In addition to looking at the rates of adaptation, we're also really interested in the mechanisms underlying these evolutionary changes. So the mutations which were driving these increases in resistance. So we sampled bacteria from all of our replicates from the end of our evolutionary treatment and sequenced all of the genomes to identify the mutations which had risen in the populations. And interestingly, we found that co-culture, that the different mutations provided the resistance when cells were growing in monoculture versus when they were growing in co-culture. So looking at E. coli when responding to, in this case, ampicillin, we saw that the E. coli largely sampled mutations associated with stress response when in monoculture, whereas in co-culture, E. coli largely sampled some mutations associated with efflux of the antibiotic. We saw a similar pattern in salmonella. There was some overlap in the genes that were sampled. However, we again saw a distinct set of genetic targets for adaptation. We were particularly interested in these mutations, which only arose in the salmonella when it was growing in co-culture with salmonella. So we repeatedly saw in multiple independent replicates, we saw that salmonella growing in co-culture acquired mutations in the protein target, so a penicillin binding protein known as FTSI. However, those mutations were never observed in monoculture. And one of the reasons we are interested in this is that many of these, in 2 of the 3 cases, the mutations were stop codons, which knocked out the function of this gene. And work by other groups in Spain demonstrated that knocking out this FTSI protein actually increases Salmonella's ability to survive inside of human macrophage cells. So growing in co-culture opened up an adaptive trajectory, an evolutionary trajectory, which caused Salmonella to not only become resistant to the antibiotic that we were selecting it with, but also drove changes in how that Salmonella would then interact with the human immune system. So we were really interested in this observation that different mutations were being selected when microbes were adapting in monoculture, co-culture. And we were wondering if that was sort of an artifact of our system or might be a more general phenomenon. And we expected or thought that it might be a more general phenomenon, because you can imagine if you start with a population of cells and you have some standing variation such that many of the cells are sensitive. You have some cells which have mutations which provide a small amount of resistance, and very rarely you have cells that have mutations which provide large amounts of resistance. In monoculture, we might expect that selection favors these mutations of really strong effect. And so that we see those mutations sweeping to fixation and dominating the adaptive trajectories. However, in co-culture, because there may be no benefit to outpacing a partner upon which you rely, selection may instead be biased by these mutations of smaller effect that are more abundant. And returning to our simulations, consistent with this idea, we saw that in monoculture, the average resistance provided per mutation was significantly higher than the resistance that was observed, than the average resistance of mutations which were sampled in our co-culture simulations. And this is additionally relevant based on some of the great work by Dr. Fuentes, showing that mutations of smaller effect may be more persistent. So the Fuentes and Pena-Miller groups did a really interesting study with E. coli in which they changed how strongly they were selecting for resistance. And what they showed is that if you apply a really strong selection for resistance, you get these big effect mutations. But then when you remove antibiotics, these mutations tend to be costly, and so they are lost in the absence of antibiotics. However, if you apply a gentler selection profile, you obtain these resistance mutations, which are then maintained even in the absence of antibiotics. We were then interested in asking whether the effects of mutations, this is a little bit of an aside, but something I wanted to get in because it's some results I'm really excited about. So we were interested in whether the effect of mutations generally may be different in monocultures or in communities. And so we developed a giant library of 300,000 knockout mutants in Salmonella. So this is a transposon mutagenesis, and the transposons carry barcodes. So the transposons insert once randomly at a position in the genome, and we have 300,000 different mutants, each with a transposon in a different location. And we can then grow this library when Salmonella is growing in a 3-species community where it's engaged in mutualism or where it's engaged in competition. And then we tested how the frequency of each of these mutants changed over the course of growth when the Salmonella was either growing alone, with E. coli, with Methylobacterium, or with both species. And we did this in a scenario where the species either were engaged in mutualism or competition. Critically, there's no antibiotics here. We're simply testing how the distribution of fitness effects changes when things are growing alone versus in more complex communities. And this was work that was carried out by 2 postdocs in the lab, Dr. Martinson and Dr. Chacon. And interestingly, we found that interaction did alter the distribution of mutational effects. So we saw that across conditions, most of the transposon insertions were neutral in all conditions, and a smaller subset of them were varying degrees of deleterious. And one of the interesting things that came out of this work was that we found that the mean fitness effect in mutualism was weaker than if Salmonella was growing alone. So you'll notice that these are, we're looking at how negative this distribution is. So mutations were far more deleterious in monoculture than they were in the mutualism. But interestingly, the average fitness effects of these gene disruptions was not significantly different across any of our competitive treatments. This buffering of deleterious effects seems to be not because the whole distribution shifted, but because a few genes which are strongly deleterious and monoculture are rescued in co-culture. And interestingly, we found that the effect of mutations in 3 species communities could be well predicted from the effect of those mutations in each 2 species community. And interestingly, we found that the fitness effect in a community was best predicted by the average effect in each 2 species community. So sorry, that was a little bit of a side, but the summaries of this point of this section are that mutualism slowed the rate of adaptation, that mutualism can alter the mechanisms of adaptation, and that mutualism alters the impact of many mutations, but that these impacts are predictable even in a community context. So finally, I want to touch on this idea. We've looked at the impact of metabolic interactions and shown that these metabolic interactions can have really dramatic effects on both the ecology and the evolution in microbial systems. And we were interested in how well we could predict that a priori. And we're at this really exciting time to be biologists, where we suddenly have this just wealth of information about genomes, where we have more and more sequenced genomes every day. But I think many of us are less interested in the genomes themselves and more interested in what they can tell us about biology. So, based on a genome, can we predict what an individual organism will eat or how it will interact with other microbes? Or another way of framing this is, can we develop tools that will allow us to go from genotype to the phenotype? And metabolism and sort of metabolic networks has a potential to provide a really powerful tool for mapping genotype to phenotype. So to do this, we developed a tool called Comets for computation of microbial ecosystems in time and space. And the way this works is we can utilize a genome scale metabolic network for multiple species. And we can then define the nutrients which are available to those species. We can then use linear optimization to optimize the physiology of each of these networks, or the flow through each of these networks, which will maximize the growth of each of those species independently. And we can iterate through multiple steps of this optimization to predict what metabolites will be consumed by each of these species, how much they will grow as a result of that nutrient utilization, what will be secreted as sort of waste products or byproducts of their growth, and then the environment gets updated between each of these iterative steps, and as a result, the secretions of one microbe can be consumed by another, allowing for facilitation. We also integrated these calculations, these optimization calculations, into a spatial lattice, and we incorporated diffusion coefficients of both biomass and metabolites. So we can start with some lattice shown here in black. You can think of this as like an agar petri dish, for example. We then seed biomass into various places on this. We can randomly distribute biomass onto the surface, and then the biomass grows as a function of the metabolites which are present in that location. And interactions, we can infer how different genotypes will interact solely as a function of the metabolism, the metabolic network encoded in each of their genomes. So these simulations generate really rich data where we can quantitatively predict the amount of biomass of each of our species through space and through time. We also get data on the concentration of every metabolite in the system through both space and time, as well as the amount of flux through every metabolic reaction in every species through space and time. And as I was saying before, we can then sort of use these additional levels of data to understand how patterns of competition or cooperation are driven by distinct metabolites or even by distinct metabolic reactions. We to experimentally test this, we first set up some experiments in which we tried to predict the rate at which E. coli colonies would expand on a variety of different carbon sources. And you can see that the observations and predictions matched up relatively well. We were particularly encouraged by this because nothing was changed about the parameter sets across any of these different nutrient environments. So we can use the exact same parameters and still get accurate predictions about the growth of E. coli in very different nutrient environments. Beyond predicting the growth rates of individual colonies, we also were able to use this tool to understand how different colonies will interact. So we were able to, for example, predict the size distribution of E. coli when it was randomly placed on a surface. And we, using the Comets tool, helped us understand that that which colonies interact is a function of both metabolism as well as of geometry, as opposed to just distance, like I had naively assumed going into it. We can also predict metabolic interactions. So we set up simulations that incorporated the 3 species I described to you before, which were engaged in cross-feeding mutualism. One of the really nice things about this is that because our models are genome scale, we can go in and introduce the exact mutation which we know our strains have. So for example, our E. coli is missing a MET-B. We can knock out that reaction in our simulations and then ask the simulation, you know, we're not defining anything else about how you interact. You know, what do you predict will be the interactions between these species? And the platform was able to both correctly qualitatively predict that no species would grow alone, but that they would engage in mutualism, engage in cross-feeding when combined. And then beyond that, it was also able to quantitatively predict the exact equilibrium frequencies of each of these 3 species. And finally, we've also been able to use it to make some predictions about the targets of adaptation. So we set up some simulations in which we went through and knocked out every gene one at a time in E. coli and grew it with Salmonella. And then looked at, again, the sort of distribution of fitness effects. And the simulations highlighted a specific knockout, which would have significant effects on the fitness in co-culture. And then in subsequent experimental evolution studies, consistent with these predictions, we repeatedly found knockouts of this gene occurred, and that the simulations correctly predicted how those knockouts would change the frequency of E. coli to Salmonella. More recently, we have released a new update of this program, and this was a significant collaborative effort that was primarily driven by Ilya Dukovsky and Daniel Segray's lab at Boston University. I should mention that the initial Comets was also developed in collaboration with the Segray lab. The new release was driven by Ilya, Georgi Bajek from Alvaro Sanchez's lab, and then Jeremy Chacon from my lab. This platform is all open source, and we've improved the documentation of it. If anybody is interested, it can be downloaded at runcomets.org. And some of the significant or the advantages of the new release include both better documentation and better interfaces. So in addition to a graphical user interface, one can also interact through the command line, through Python, or through MATLAB. Some additional biological complexity was incorporated through incorporating demographic complexity, such that and improved biomass spread. So instead of just predicting perfectly circular colonies, we can now get these really interesting dendritic patterns arising as populations expand. We've also incorporated extracellular enzymes so that in addition to intracellular metabolism, it's now possible to incorporate the production and the dynamics that arise through the release of enzymes that have activity outside of cells. We've introduced both mutation and evolutionary dynamics. So what you're seeing here is time on the x-axis, biomass on the y-axis, and these were randomly introducing mutations into the system. And you can see that mutations accumulate and you start to get interesting clonal interference between different mutants within our population. Finally, we've significantly increased the environmental complexity that can be modeled. So for example, it's now possible to model a slice of the soil where you might have an air interface at the top, some nutrients released by a root, and then various barriers to diffusion, such as rocks or soil particles within your simulation. And we can make some really interesting quantitative predictions about, where biomass will arise and the nutrient gradients that will arise as a result of microbial activity. I'll also highlight that these simulations are capable of incorporating hundreds of species at a time. And so we think that they may provide some useful tools for starting to make some quantitative predictions, even in very complex environments. And I'd also like to highlight that I think that this could be a really powerful complement to some of the great work that has been developed in the Resendus lab, where they've worked with MICOM, which is another system where you can use genome scale metabolic models and incorporate metabolic models of multiple species into a single simulation. And some of the powers of their system are that they constrain the maximum growth rates that are possible of individual models. And they've shown that this approach can be really powerful for making some strong predictions about the types of interactions and the community function of complex microbiomes. And so I'm really excited to try and dig in and think about how these two different approaches can complement each other and when and where they will make distinct predictions. So I hope that's given you some sense of some of the ways that we're starting to take a bottom-up approach to microbial communities. And in conclusion, we've shown that metabolic interactions alter inhibition and persistence, that metabolic interactions alter the rate and mechanisms of resistance evolution, and that metabolism can be used to predict community dynamics. I'd like to thank some fantastic collaborators and lab members, as well as some of our funding sources. And with that, I'd be happy to take any questions.
Thank you, Will. So for questions, please raise your hand and open your microphone. And the people who is following in YouTube, please send your questions by the chat. So I don't see, yeah, Alberto Pantoja, please go ahead.
Hi, first, Daniel Machado from NTNU sends you greetings. And my ask is, in the model, how is the degradation of polymers such as alginate or cellulosa result? Sorry, I got nervous.
Sorry, I'm not sure I totally follow that. So your question was, how are polymers,
yeah.
How are they degraded?
Yeah. In the model.
In the model, how are,
yeah.
When that's done extracellularly, I mean?
Well, in a work, I was doing simulation of biosensor, and I was working with polymer, and this, when I want to cut, I don't know how to represent it in the model because the size of it was undeterminate, so I don't know how to represent it.
So you have a polymer, but it's unclear exactly what the chemical composition of that polymer is, and so the question is, how would you incorporate that into into genome scale metabolic modeling?
Yeah, like PET.
Yeah. So that is hard, and that's certainly one of the constraints about this approach is that it requires a distinct characterization of all the metabolites that are present. As a first pass, one can introduce a novel metabolite, so just sort of a fake metabolite, and then one can make some inference about the amount of energy that could be acquired from that metabolite. And one could test many different possible sort of nutrient environments or many different possible amounts of energy that a cell could get, and then compare experimental observations against those different simulations, and potentially use something like that to get some better estimates of how much energy or other nutrients cells are acquiring from the polymer, from sort of the unknown polymer. Does that? I think I'm not exactly answering your question, but does that sort of provide a starting point at least?
Oh, I don't have it right now.
Okay. Well, yeah, I'd love to follow up with you further, maybe through email, or maybe we can okay. Talk some more. Yeah, I would definitely like to understand a little bit more, and it'd be fun to think through it.
Thank you very much.
Thank you.
Another question? Yeah, go ahead, Rafael.
I will. Well, more than a question, I was just kind of like, I want to say hi. But I was wondering if you have implemented antibiotics in Comets, because it seems like you have already kind of like small molecules diffusing. So I was wondering if you have some sort of implemented inhibitors to the metabolic network.
We do, and yeah,
I probably missed it.
No, we did. I didn't mention it. That's a paper that's on my desk that we're hoping to get out soon. But yeah, you're exactly right. So we've implemented toxins as well as actually signals. So we have the potential to incorporate quorum sensing, and exactly like you're suggesting, both toxins and signals have they're metabolites that have concentration-dependent effects on the physiology of microbes. And so we track the concentrations of them as they're released from cells, and then have encoded it such that those toxins either put constraints on the death rate or on the growth rate of our models, and that signals can alter they can either shut down specific reactions or force more flux through specific reactions. So we've at this point, we're using it to look at when toxin production is effective. But I think there's lots of really fun things that could be done looking at antibiotics more generally and how those will influence communities. And
I agree. Yeah. I agree. Absolutely agree. There's even maybe that first example that you showed us when you have like some sort of density. I mean, you had more persistence when you had like. Because even in your little drawings, you would see like when you have less cells, you have more distance between them.
Yep.
So therefore, the strength of the interaction would probably change, and then their susceptibility.
Yeah,
I don't know.
yeah.
It seems like you could test a lot of things with that model.
Yeah, I think, that is definitely a direction we're excited about going. It's, some of the individual heterogeneity stuff is a little bit harder just because we're not modeling individual cells. It's not agent-based. But I think we could still capture some of it. That is definitely a direction we're excited about going. And then additionally, there's more and more data on the ways that antibiotics alter the metabolism of cells, in addition to just sort of killing them. And so we're excited about trying to incorporate some of those effects into the simulations as well, to get better predictions about how antibiotics will alter, again, the composition and function of communities. But yeah, it's still definitely early days.
Oh, but it sounds exciting. Well, nice to see you.
Good seeing you.
There are two questions in YouTube from Cesar Ayala. He asked, could Comets work with complete metagenomes files? That is Binix, or is preferably to have individual genomes as inputs?
Yeah. Let's see. Where I think it would work best is if you could do a MAG, so a metagenome assembled genome. So if you could assemble individual genomes for the different species in your metagenome, there I think the power of Comets would be strongest. Another approach that one can take is to sort of treat the community like just a bag of genes. So to take all of the genes that one observes in a community and put them into a single model, and then make some predictions about what pathways are feasible, incorporating the activity of all of those cells. I think that one of the powers of Comets is that it optimizes the growth of individual cells. And so to get the dynamics that would come out of that, I think one would need individual models for each of the species you are interested in.
Okay, thank you. His second question is, how can one validate the prediction model of a microbial community?
So there are lots of predictions that come out of the simulations. One of the easiest things to test, and one of the ways we often go, is to look at to test the predictions about the population size. So how population size changes through time. So in many of our systems, we have our individual bacteria labeled, and we can quickly measure in real time the abundance of each of them. But one could also compare simulations against, for example, abundances which were acquired from metagenomic sequencing. Beyond that, one could sort of make comparisons from the model against either metabolomic or transcriptomic data. So if you had either of those, the model makes predictions about exactly which metabolic pathways will be active or which metabolic enzymes will be expressed. And so one could compare at that level. And then again, at the metabolomic level, there are predictions about the concentrations of every metabolite in the system. And so one could, one could compare against those sorts of properties as well. Or alternatively, if there's a function of interest, so how quickly carbon is degraded or how much total biomass is produced. Those are all predictions that would come out of the model as well.
Okay, so is there any question in Zoom? So they can ask it right now, or I continue reading the questions from YouTube? Okay, if no questions by Zoom, so the next question is by Luis Alcaraz. He says, fantastic talk. So it will be possible to model any couple of genomes to predict dynamics. What are the minimum requirements for inputs regarding metabolites, genome completeness, for instance, max?
Yeah, so for Comets, the inputs are metabolic models, so a metabolic network for each of your species of interest. And then you have to define the metabolites that are present in the environment. One certainly can put in sort of very simplified networks. So, you know, you could create sort of cartoon models that have maybe two reactions that showing cells using different metabolites or you know, any number of scenarios. The more information one has on the more complete the networks are, the richer the predictions that come out of it are. There are pipelines which have been developed, you know, other great pipelines which have been developed for taking, if you have a genome sequence, you can then put that genome sequence into this pipeline, and it will then take, based on the genome sequence, an annotated genome sequence, it will identify the metabolic enzymes that are present in that genome, and then convert those enzymes into reactions and generate genome scale metabolic networks for you. So CarveMe and Raven are two popular tools for generating new models from genome sequence. Additionally, there are some fantastic databases out there that already contain many metabolic models. So the big database has 100 sort of manually curated models, and then the Agora 2 now has 7,000 so genome scale metabolic models for 7,000 unique organisms, and I think KBase has upwards of 10,000. So yeah, there's sort of multiple ways to find or generate models, and then once you have those, yeah, you just need a model for your species of interest and the metabolites that you're interested in, or sort of the metabolic environment that you're interested in modeling them in.
So next question is from Diego Montes. By saying that metabolism is an intermediate point between genotype and phenotype, would this mean that metabolites are epigenetic modifiers in the entire community of microbes?
Does this mean that metabolites are epigenetic modifiers in entire communities of microbes? I guess I think of epigenetic factors as being things that are heritable that are not genetic in basis. So to the extent one is thinking about, you know, metabolites are a way that microbes can modify their environment, which can alter the behavior of additional cells. So yeah, I guess I think that potentially one could think of them that way. Yeah, I mean, I think that the metabolites are certainly very important drivers of interactions between cells and drivers of emergent community properties. So yeah, I think I would certainly agree with you in that context. Yeah, I hope that I answered some of your question. I'm not sure I fully got it.
Leonor Perez asks, could comets predict changes in the metabolism of the communities in response to prebiotics or in disease conditions?
Yeah, absolutely. So prebiotics is one of the things that it should be particularly great for. So you can set up simulations where you start with some community and then predict how, you know, the species frequencies or the function of that community will change across thousands of different metabolic environments. So for example, one could use it to identify novel prebiotics, which would change the composition of the community in desired ways. So that would be one powerful way to go about it. Or disease conditions. And yes, I think it could also be used to look at disease conditions. So for example, in MECOM, which is a similar approach with some slightly different assumptions, they were able to identify some of the metabolites associated with diabetes, I believe it was. And so how the metabolites and the composition of the communities changed in healthy states versus disease states. So yeah, I think both of those things should be possible using genome scale metabolic models and different programs to look at them.
Okay, so another question by Alberto Pantoja is regarding the spatial capabilities of the model. Can we also make simulations of bacteria in microfluidics chips?
Yes, yeah, you should certainly be able to do that. And so it's possible to model sort of well-mixed liquid situations, or to modify or to model any variety of different spatial configurations. I will say that it works best if you have a 2D scenario. So 3D scenarios, there is a 3D version of Comets, but it doesn't work quite as well. So if you can take a 2D slice, but particularly in microfluidic devices, that should be a good approximation of what one is seeing. Again, I'll highlight that Comets is not actually simulating individual cells. It's instead simulating sort of populations of cells. So to the extent that the dynamics you're interested in are really driven by the behavior of individual cells, like they sometimes are in microfluidic devices, some of those behaviors might not be as well captured. But I think that it should, it certainly, so the scale of the Comet simulations, one defines the size of the pixels in your lattice, and that can scale from micrometers to miles. So it should be feasible to run simulations across many different scales of size scales. So yes, I think that in theory one could certainly simulate microfluidic chips.
Any other question? Well, I have one, Will.
Yeah.
Regarding your Salmonella mutants, have you actually tested their ability to survive in macrophages?
No, we haven't done that yet. We are working on those measurements ourselves with some collaborators here, but we haven't. We haven't tested our mutants specifically in macrophage yet. So there we're relying on the published data by other researchers showing that knocking out the gene, which we see knocked out, increases survival.
Okay, that would be interesting.
Yeah, I'd love to do that. Yeah, we don't quite have the expertise yet, but.
Okay. So if there is no further questions, thanks again, Will, for your seminar. Very interesting data. And already people is asking me for your email, so I will send it so they can contact you for further questions and more in detail. Whatever they want to ask. Thank you again for the lecture with the students and for the seminar. And hopefully Excellent. In the future we will see you here.
Yeah, that'd be fantastic. I would love to come down. So.
Okay.
Excellent. Thank you, everybody.
Thank you, everybody.